Hey everyone, you can turn on the audio. Sorry about that. So today we're going over signal transduction. Um, yep. So I'm gonna have the chat open. So if you have any questions, just type in the chat or just speak out loud. If I'm George, Lily's also here. Yep. Okay. And this will be recorded too and posted on the YouTube channel after, so you can look at that, and we will answer questions at the end. Okay, so part one, we're going to go over the principles of signal transduction. So this is covering the first half of the week of videos that Laura posted. So firstly, we have signals. So signals are usually small molecules. Um, and yeah, so by small molecule, it's the size of one or just a few amino acids. So I think like, I don't know, like glutamate by itself would count as a signal, I guess, or could. And then there are also proteins, which are larger. Those won't be counted as small, but those can be signals too. Um, so signals are either hydrophilic or hydrophobic. So hydrophobic signals can pass through the membranes or pass through the membrane, well, because of the phospholipid bilayer. The middle is hydrophobic, while the outside layer is hydrophilic. And then, so there are large and small hydrophilic molecules are not likely to pass through the membrane. I'm sure there are like some situations where they can, but for the purposes of this class and just in general, they probably can't pass through the bilayer, regardless of their size. And the cell also has receptors on the surface to transduce information from hydrophilic signals. So it transduces information from the signal. The signal itself just binds to a receptor and doesn't go through. That was a pretty common misconception, I think, from last semester. So yeah, the signal binds to the receptor, causes conformational changes in the cell, which is how it makes an impact. So it leads to more and more effects as it binds to the signal. And we'll talk about how that happens. And so here's a question. What type of signal is best able to pass through the cell membrane? Specifically, whether it's hydrophobic or philic and its size. And if you don't have an answer, you can like talk or type it in the chat. Yep, that's right. Small hydrophobic with DCs. That's perfect. So yeah, this was on one of my exams at least, where it asked like what is able to get through the membrane, and I remember I was really torn between like large hydrophobic or small hydrophobic because they're both hydrophobic, but it's specifically the small hydrophobic molecules that are best able to go through the cell membrane. I'm not sure exactly if large ones can, but not as well as small ones, and that's the important part. And also, yeah, there's nitric oxide in the top right corner as a small hydrophilic one, which probably cannot get through a membrane, but it can bind to a signal or a receptor, I mean. So next we have outcomes of cell signaling. This one she went over in class too. So there are two outcomes: change in protein function or just change in protein synthesis. And main thing to know is that they're not mutually exclusive, and that while a change in protein function is faster, sometimes like the cell doesn't need it to be just faster, but it also needs like protein synthesis to have like a more like lasting effect. So insulin's a really good example of this. I believe she talked about it pretty extensively in the video too. And so both outcomes, as you can see in the chart, just lead to altered cell behavior. And that's the outcome of both. Yep. And that's basically it for this part. So here, something good to know is that cells can receive multiple signals at once. So it's not just like the one signal. And it is easy to like fall into that like that sort of like mindset, because when we talk about like signaling, we talk about this pathway or this signal leads to that, but a cell like at once, like a lot of signals are happening and going on at the same time. Yep. Something in the chat. Oh, never mind. Okay. Yeah. So for example, the survival signal pathway, which we'll talk about later, I think later in the week. Like it's always receiving survival signals in order to live. So like when you see a cell receiving like a signal to divide or to differentiate or to do like whatever, it's usually also receiving a signal to survive, because for that signal, its automatic pathway is to die. Um, Let me check the chat for a second. Okay, yeah, okay. Yep, so as you can see in the picture here, it's receiving multiple signals telling it to survive. and it's always receiving that, well, regardless of whether it's like dividing or differentiating. And once it doesn't receive that signal, it just dies. So that's an example of how like a cell is always receiving more than one signal at once. So for the first question, what happened if you isolate a cell away from the body? You can answer in the chat again, or just say it out loud. That's perfect, Maya. So if you take it away from the body, it won't be receiving the survival signals anymore, and it will just like die. Because that's what would happen. So on the flip side, what would happen if there was a mutation that caused survival signals to be permanently bound to the cell? So the signal just like stays in the receptor forever. That was a mutation. There was an extremely like absurdly high affinity. What would happen in that case? There's like a specific term for it. That's used pretty often. It's like a disease. Yep, that's right. Cancer. Yep, and that's pretty much it for this. So the same signal can lead to different outcomes. So obviously what type of like receptor it is or what type of cell is receiving the signal, it can lead to a different like effect, even if it's the same signal. So it's just because like each cell or each protein expresses different proteins and different genes, and each one is unique. So even if it receives the same signal, it can have like a different reaction. And the term LoRa used specifically is like downstream or molecules and proteins downstream to the receptor. So even though the receptor is the same because it's binding the signal, the rest of the cell isn't. And you see it on an exam or like I don't know in your problem set, writing downstream to the receptor is probably like a good thing to write because it's a keyword that LoRa is probably looking for. Yep. And then signal amplification. So starting with the signal binding to a receptor, it can communicate with multiple downstream components. And those downstream components can communicate with even more downstream components, and that leads to an exponential increase in the signal. So if you can see here in the picture, the signal binds, it communicates to many CAMP, which communicates only just one PKA, but then each of those PKAs can communicate with multiple of the target protein. So if you think about like a cell signal binding to one thing, the receptor only interacts with one like downstream molecule at a time. It will take like forever for anything to happen, for any pathway to happen, which is just like not efficient or good for a cell to live. So that's why signal amplification is important. So first question, should we expect signal amplification in longer or more signal amplification in longer or shorter pathways? What do you guys think? Yep, that's right. So the longer the pathway is, the more chances there are for it to amplify other molecules. While like a shorter pathway, if we just went to the signal to like the CAMP, that wouldn't be as good like here to like the entire thing. So being longer is usually when there's greater amplification. And crosstalk. So different pathways can communicate with each other. So you can just see here, like yeah, just looking at the picture, it's a little bit messy just looking at it. But if you look at the survival factors and the RTK, that's like a survival pathway. You can see like the GPCR, the G proteins interacting with it and affecting it. And then the importance of this is that it can just regulate like other pathways, different functions. It makes the cell more efficient and organized, which is like a pretty common theme that the cell is very efficient and organized. So similar question to the last one, do you expect crosstalk to be more common in longer or shorter pathways? What do you guys think? Yeah, that's right. It's longer. So just like the main point of like these questions is just that like the longer the pathway, obviously it's longer and it's probably going to be like it's going to take more time than the shorter pathways, but there are more chances to regulate it via like amplification or crosstalk. Yeah, this is the point of having a longer pathway versus a shorter one. And then I think that's it for me. So Lily can take over. I turned off my volume so you can talk.
Perfect. Can you guys hear me? I'm not on my regular computer, so.
I'll look at the chat.
And then could you just stop sharing your screen so I can turn my.
Okay.
Okay. All right. Can I hear?
Stop your plane. Turn it off.
Oh, okay. Yeah.
All right, good.
Okay, you can hear? Okay. All right. So I am covering the RTK RAS MAPK pathway. This is the only pathway, like actual pathway, that we're going to cover tonight. And I think the Thursday group is going to do the others just because this is like a kind of dense topic. So just as a quick overview, the RTK RAS MAPK pathway is found in eukaryotes. The examples that Laura gave were Drosophila, budding yeast, in humans. And this specific pathway is primarily involved in division and differentiation signaling, but RTKs just in general can do a couple more things like insulin signaling and survival signaling. So just as a quick review, kinases are enzymes. They're a type of protein that phosphorylates their substrate, which is usually also a protein. And that's a type of post-translational modification. They do that by taking ATP, cleaving the bond between the second and third phosphate, and then attaching the phosphate to an oxygen on their substrate. And usually the effect that has on the substrate is that it acts as like an on switch, but not always. So here's like a little true or false question. You can put an answer in the chat or unmute your mic. Is it true that because post-translational modifications are covalent, phosphorylation is irreversible? Oh yeah, Maya, false. Do you guys want to put why, or I can also explain? I don't want to put you on the spot. Okay, I'm gonna move to my next slide. So, oh, answer. Right, yeah, so you can dephosphorylate with phosphatases, which essentially just reverse that, and they take the phosphate off, and that's more like an off switch in most cases. Nice job. Okay. So, oh, one of my, okay. I don't know why that did that, but so there are a couple of classes of kinases that we see in eukaryotes. One that we're going to talk about tonight is tyrosine kinase, and the others are serine threonine kinases. And the thing that they have in common is all of their R groups have a terminal hydroxyl, and that allows the little phosphate group to attach when they're carrying it from the ATP to their substrate. Okay, so to get into the actual pathway, I think you guys can see my mouse, so I'm going to point with that. See the mouse? Yeah, okay. Yeah. So basically, we start off with two RTK inactive molecules, and then the signal comes in the form of a dimer. So that a dimer is basically when two proteins or molecules or whatever come together and form their own functional unit. So they dimerize, they bind to the receptor domains of the tyrosine kinase or RTKs, and this causes them to slide together and stimulate their kinase activity from these little tyrosine kinase domains. So when that happens, they can autophosphorylate, and these phosphate groups that we have on our active RTK are binding sites for downstream proteins. So this is just showing here that you can have like multiple proteins binding because we're phosphorylated in multiple places. But this blue one at the bottom is the one that we're going to be focusing on. So quick question before we move on. If the RTK molecules are embedded in the membrane, how is it that when the dimerized signal binds, they're able to slide closer to one another? And this is, I guess, a little bit of a like 151 review. I can go back to the diagram. So when this signal binds, how did these come together? I'll give it a couple more seconds. Oh, Carla knows. Okay. Oh, does Carla know? I don't know if Carla knows, but I'll just put up the answer. So basically the thing that allows those RTKs to slide around is the fact that the membrane is fluid. So this is something that we talked about in 151. Either, having like unsaturated lipid tails or heat, in the membrane gives it a certain amount of give where stuff can kind of slide between those hydrophobic heads and move around when they need to. So you have that lateral diffusion happening. And this right here, these little gray lines are our cell membrane. So when this dimerizes, these kind of just diffuse laterally through the membrane. And then one little review question also, because this is going to be relevant for the next step. What is a GEF? What does it do? You guys should remember this from the exam. But if you don't, it's okay. All right, Carla. So GEF facilitates the exchange of GDP for GTP. And basically, like when it binds, it causes the GTPase, which in this case is going to be RAS, to lose affinity for GDP. And then that open binding pocket allows GTP to diffuse in. So we're going to see that in a sec. And then just some more context before we get started. PXXP motifs and SH3 domains. So we talked, I think, maybe in topic 1 about how the amino acid proline is pretty uncommon in alpha helices. And that's because we have this internal ring structure here. So that makes it rigid, and you don't usually find them like in the middle of that twisted helix because it just kind of throws off like the geometry. So there are some cases where an area in your primary sequence of amino acids where you have a heavy concentration of proline, that can form a polyproline helix. And PXXP motifs are recognized, or the binding substrate, I guess, for SH3 domains, which we're going to talk about. And PXXP motifs often contain polyproline helices. So little tidbit. All right, so once we have our dimerized signal, our dimerized activated RTKs, we have this little adapter protein, that's what they call it on the diagram, but I think we're mostly going to call it GRAB2, that binds to the phosphate group with its SH2 domains. And SH2 domains, I think we see in one other pathway, they're specifically a phosphotyrosine binding domain. So they are always going to bind like this, essentially. And then the other side of GRAB2 has an SH3 domain, and that is what is going to bind the PXXP motif and our RAS activating protein. So RAS activating protein functions as a GEF. It might also be called SOS, son of sevenless. We talked about that, or Laura talked about that in the video. But what that is going to do is it's going to, I guess, reach up to our inactive RAS. It's going to act as a GEF. It's going to help GDP leave the binding pocket. GTP is going to diffuse in, and then we have active RAS. Is everybody following so far? I don't see any questions in the chat, so I'm going to keep moving. Okay, so then this is the last part of our little pathway. So our activated RAS, which was activated by that RAS-GEF, helping it exchange GDP for GTP, is going to activate MAPK3. So a really common misconception is that RAS phosphorylates MAPK3, but RAS is not a kinase. So we don't know exactly how it activates it, but it does, and that's all you need to know. So when our MAPK3 is active, then it's going to activate MAPK2, it's going to phosphorylate it in 2 places, and then MAPK2 will activate MAPK, also phosphorylate in 2 places, and then MAP kinase can go and basically, depending on like what the initial signal was, what the pathway is aiming to do, it can, go and phosphorylate either a downstream effector protein or a transcription factor regulator, whatever. And what it ends up phosphorylating will vary depending on what the signal was. So like if it was a differentiation pathway or a division pathway, whatever, it might phosphorylate something different. So we have these 3 MAP kinases here. It seems kind of redundant that we would have all of these in this kinase cascade, but what would be the benefit of this redundancy? What purpose would that serve? Yeah, amplification. So basically with all of these steps, our MAPK3 at the very top here, let's just say it can go and these aren't actual numbers, but let's just say it goes and phosphorylates 5 MAPK2s, and then those 5 MAPK2s activate 5 MAPKs. Then, this one little signal from our activated RAS has a much stronger response from the cell. So that would be the benefit of this kind of redundant pathway here. Okay, so then I just kind of put all of these together following the arrows. This is something that really helped me because this is kind of a big diagram. There's a lot of stuff going on. So just to see it all in one place, this might be just thinking way ahead, something that you put down or draw out on your next note sheet for exam 2, just so you can keep all of these straight. I think it's helpful to see them all together. So, and then here is just a little overview. I'm not going to go over this because this is what we just talked about. But if anybody has any questions on this pathway in particular, we can go over that. This is the first time we kind of did pathway stuff with Triboperon, but I think this is a little bit unfamiliar. So we can slow it down. We can go back over any of those steps if you're confused, or we can just take some questions, maybe do some problem set questions after we stop the recording. So I'll give you guys a minute to type, or you can just give a thumbs up if you're all good. All right, all good. Awesome.